Hi everyone, my name is Begita Misura, and in this short presentation, I want to talk about our work, which we call DL Packer, deep learning for prediction of amino acid side chain conformations in proteins. As you probably know, proteins are a kind of biomacromolecules and one of the fundamental building blocks of life. They do just about everything inside living cells. They serve as catalysts to speed up reactions, they are structural components of cells, they transport various cargoes and transmit signals, to name just a few of their roles. This variety of functions leads to a variety of shapes and structures, as illustrated on this slide. In this work, the task we're working on is to restore amino acid side chains starting from fixed backbone coordinates, the task which is also termed packing. What is important for this work is that conceptually proteins can be viewed as a sum of two parts, backbone and side chains. And what we want is to restore the side chains given only backbone conformation. This can be useful in many ways. For generation of full atom structures from reduced representations, refinement of protein structures. As one step of protein structure prediction, protein design, and so on. In this work, we trained a deep 3D convolutional neural network to solve this problem. I will not discuss any details right now, but feel free to reach out to me during the process session with any questions. I will be happy to dive into details. As a result, our model, which we call DL Packer, significantly outperforms other available methods for packing, as illustrated on this slide. This graph shows root mean squared deviations of atom coordinates for each kind of amino acid in proteins. The lower is better here, so DL Packer outperforms other methods for each and every kind of amino acid. We have also analyzed RMSD histograms in detail and found that DL Packer yields significantly lower rate of high RMSD predictions, as shown on the left, both on the surface of proteins and in the interiors, as shown here on the right. Please reach out to me if you have any questions during the process session, and I will be happy to discuss our methods and the results in much more detail. Hope you found this interesting. Thank you.